In the cell, you can imagine DNA like royalty. It sits in the nucleus, and that's where it directs the activities of the cell. When it wants something to be done in the cell, enzymes to change, receptors, whatever needs to be done in the cell, or something that needs to be exported like antibodies or whatever, then the DNA will send a messenger to get the job done. So the information from the DNA will be transcribed in the nucleus into a messenger called messenger RNA. It is the messenger that will leave the nucleus, go into the cytosol, and have the message on the messenger RNA translated into a way that the cell can understand and do something, do some sort of activity. And that translation will be into a protein. And the proteins are the workers of the cell. So let's look at the story more specifically. To begin with, the information on the DNA, which will be transcribed and then translated into a protein, is called a gene. I use the word protein, but it also stands for polypeptides as well. A polypeptide is exactly like a protein, but much smaller. Now, of course, we've seen that the DNA is a double helix. The RNA molecules are single-strand molecules. And this is being produced using DNA as a template, transcribing the information. The information is not word for word for a couple of reasons. One is that we've already seen that RNA writes its messages differently than DNA. Uracil is the base in RNA as opposed to thymine, and furthermore, the sugar is ribose as opposed to deoxyribose. That's one issue. The other issue is all the information in a gene on the DNA is not necessarily useful information. Some of this information would be called introns. It's not expressed. The information which is expressed, exons, so those are expressed information that will be translated into a protein. So the RNA will have to take all the information from the gene and then will have to start maturing itself, removing the introns, which are not important, and keeping the exons, which will be translated. So once the messenger is created, then it will leave the nucleus. So we imagine some sort of single-strand molecule, which is the messenger. So the messenger leaves the nucleus, goes into the cytosol, where its information will be translated. In order for it to be translated into a protein, it needs the proper environment. The environment for translation is provided by a ribosome. A ribosome is a complex of proteins and what is called ribosomal RNA. We mentioned earlier that ribosomal RNA is concentrated in the nucleolus of the nucleus. So ribosomal RNA and proteins will complex in the cytosol or associated with the rough endoplasmic reticulum, which is studded by ribosomes. And so this complex called a ribosome, which is rRNA and protein, will provide the necessary environment for the mRNA to be translated. Now, when we look inside and we see the messenger and the environment, what we will see on the messenger is a code. The code for genes is a triplet code. That is called the genetic code. And so when we look up closely, we will see on the messenger RNA some sort of triplet code of 3 nitrogen bases. For example, it might say AUG. So AUG might be the code that shows up within the ribosome. So in other words, there's the translation that will be taking place from the messenger RNA into an amino acid, which is the building block for a protein, will come from the genetic code, which is a triplet code. So AUG is present here. That code on the messenger RNA is called a codon. So we have the code, we have the environment, now we just need something that will translate this code into an amino acid and take that amino acid and transfer it onto another amino acid and another amino acid, etc. And all these amino acids linked up together will form our protein. So the molecule that will be able to read the codon, transfer an amino acid is called transfer RNA or tRNA. So we imagine tRNA as a molecule that will have one part which will be able to identify the codon and another part that will be carrying an amino acid. This part that will identify the codon will be called the anticodon because it will bond to the codon. So this would be 3 bases that would identify this particular codon. And these 3 bases will always come with this particular amino acid. So you may want to pause here and consider what would be the names of these 3 bases on the anticodon which would identify this particular codon of AUG. Well, the complementary bases for AUG would be U, A, C. The reason is because we saw that in DNA, A bonds to T, G to C. But we also said that thymine is replaced by uracil in RNA, and these molecules are RNA molecules. mRNA, there's tRNA, and of course the environment is in part prepared by rRNA. So these are RNA molecules. So T is out of the question and we're only talking about U. So then A will bond to U instead of T, and then U will bond to A instead of T to A, and G bonds to C. So UAC is the anticodon which would attach to AUG. And when it comes in, it attached, it leaves off its amino acid, it goes away, the ribosome moves over, reads another part of the genetic code, another triplet of nitrogen bases, another transfer RNA will come in, read the codon, bond with its anticodon, release an amino acid, transfer it onto the polypeptide, which is growing into a protein. So that is how the translation occurs. Now, in terms of the protein, the subunit of proteins are amino acids. And amino acids are called amino acids because on one end there is an amino group, which is the NH2 group, and on the other end there is an acid group. And of course, when you talk about organic chemistry, an acid group in organic chemistry is a carboxylic acid. So it's COOH. So that is the acid group which is on one side. So it so happens that as our polypeptide is being produced and getting longer, on one end there is going to be the amino group. That's the first thing that comes out. Then in the amino acid there's the carboxylic acid group. That will bond to the amino group of the next amino acid. The bond between that carboxylic acid group and the amino group, that's called a peptide bond. So the peptide bond produces the polypeptide, which produces the protein. So then at the beginning of the polypeptide, you will have one amino group sticking out. Then when you go all the way to the end of the polypeptide or protein, you will have the carboxylic acid group that will be sticking out at the other end. So that is how the protein is being produced. Now, if the ribosome is just free in the cytosol, and it meets with the messenger RNA and the transfer RNA, and this reaction is occurring and the protein is being produced, then this kind of protein might be used within the cell for some cell purpose like enzymes within the cell, etc. If this happens to be a ribosome, which is studding the rough endoplasmic reticulum, then this protein is being pushed into those flattened sacs of the rough ER. And then after it is produced by the rough ER may send it to the Golgi apparatus where it will be further processed and then exported to the cell membrane. That is where those proteins might be used within the cell membrane itself or might be exported for other purposes, for example, antibodies or a protein enzyme.